Hi everyone, this is Dr. Udwak Lectures. Today we'll be talking about hemoglobin. Hemoglobin is the iron-containing coloring pigment of red blood cell. It is actually a chromoprotein that is responsible for carrying respiratory gases such as oxygen and carbon dioxide, and it also acts as a buffer. The average hemoglobin content in blood is between 14 to 16 grams per deciliter. The values actually varies depending on the age and the sex of an individual. At birth, the hemoglobin content is 25 grams per deciliter. After the third month, the hemoglobin content is 20 grams per deciliter. After a year, the hemoglobin content is 17 grams per deciliter. From puberty onwards, the hemoglobin is between 14 to 16 grams per deciliter. Now, at the time of birth, hemoglobin content is very high. It is high because of the increase in the number of red blood cells. Now, in adult males, the hemoglobin content is 15 grams per deciliter, and in adult females, it is 14.5 grams per deciliter. The main functions of hemoglobin is the transport of respiratory gases. Hemoglobin transports oxygen from the lungs to the tissues. It transports carbon dioxide from the tissues to the lungs. When oxygen binds with hemoglobin, a physical process called oxygenation occurs, resulting in the formation of oxyhemoglobin. When carbon dioxide binds with hemoglobin, the carbohemoglobin is formed. Now, the affinity of hemoglobin for carbon dioxide is 20 times more than that for oxygen. Hemoglobin also acts as a buffer and plays an important role in the acid-base balance. Now, hemoglobin is a conjugated protein. It consists of a protein combined with an iron-containing pigment. The protein part is called globin, and the iron-containing pigment is called heme. Now, iron normally is present in hemoglobin, but then it is present in the ferrous form. The ferrous ion is an unstable or loose form of iron. The porphyrin is the pigment part of heme. It is usually formed by 4 pyrrole rings. The globin usually contains 4 polypeptide chains. Now, among the 4 polypeptide chains, 2 are alpha chains and 2 are beta chains. Hemoglobin is of 2 types. Number 1 is the adult hemoglobin. Number 2 is the fetal hemoglobin. In adult hemoglobin, the globin contains 2 alpha chains and 2 beta chains. In fetal hemoglobin, there are 2 alpha chains and 2 gamma chains instead of the beta chains. Now, functionally, the fetal hemoglobin has more affinity for oxygen than that of adult hemoglobin, and the oxyhemoglobin dissociation curve of fetal blood is shifted to the left. Abnormal types of hemoglobin or hemoglobin variants are the pathologic mutant forms of hemoglobin. These variants are produced because of structural changes in the polypeptide chains caused by mutation in the genes of the globin chains. Now, most of the mutations do not produce any serious problems. Number 1 is hemoglobinopathies, and number 2 is hemoglobin in thalassemia and related disorders. Hemoglobinopathy is a genetic disorder that is caused by abnormal polypeptide chains of hemoglobin. Some of the hemoglobinopathies are hemoglobin S. Now, hemoglobin S is found in sickle cell anemia. In hemoglobin S, the alpha chains are normal and the beta chains are abnormal. Hemoglobin C, the beta chains are abnormal. It is found in people with hemoglobin C disease, which is characterized by mild hemolytic anemia and splenomegaly. Hemoglobin E, the beta chains are abnormal. It is usually present in people with hemoglobin E disease, which is also characterized by mild hemolytic anemia and splenomegaly. Now, hemoglobin M is an abnormal hemoglobin that is present in the form of methemoglobin. It occurs due to mutations of genes of both alpha and beta chains, resulting in abnormal replacements of amino acids. It is present in babies that are affected by hemoglobin M disease or blue baby syndrome. It is an inherited disease that is characterized by methemoglobinemia. Let's talk about hemoglobin in thalassemia and related disorders. In thalassemia, different types of abnormal hemoglobins are present. The polypeptide chains are decreased, absent, or abnormal. Now, in alpha thalassemia, the alpha chains are decreased, absent, or abnormal. And in beta thalassemia, the beta chains are decreased, absent, or abnormal. The synthesis of hemoglobin begins in the proerythroblast and continues even into the reticulocyte stage of the red blood cells. Now, let us look at the basic chemical steps in the formation of hemoglobin. First, succinyl-CoA, formed in the Krebs metabolic cycle, binds with glycine to form a pyrrole molecule. In turn, 4 pyrroles combine to form protoporphyrin 9. The protoporphyrin 9 then combines with iron to form the heme molecule. Finally, each heme molecule combines with a long polypeptide chain known as the globin, which is synthesized by the ribosomes, forming a subunit of hemoglobin called hemoglobin chain. Now, 4 of these in turn binds together loosely to form the whole hemoglobin molecule. Now, after the lifespan of 120 days, the red blood cell is destroyed in the reticuloendothelial system, particularly in the spleen, and the hemoglobin is released into plasma. Soon, the hemoglobin is degraded in the reticuloendothelial system and split into globin and heme. Globin is reused to synthesize hemoglobin. Heme is degraded into iron and porphyrin. Iron is stored in the body as ferritin and hemosiderin, which are reused for the synthesis of new hemoglobin. Porphyrin is converted into biliverdin. In humans, most of the biliverdin is converted into bilirubin. Bilirubin and biliverdin are together called the bile pigments.